Yeah, okay. I'll put it somewhere.
Issue solved. Well,
Here.
our speak is Andrea de Martino. He will be talking about RNA crosstalk
this
at
way.
system level.
No, not yet. What?
Okay, kill it. Kill this one. Kill this one. Kill. Kill this window.
Kill this window. Okay. Is it working?
Yes.
Cool. So this goes. Okay.
And the pointer.
So sorry about this. Right. So thank you very much for having me, for organizing this. It's great to be here, and I'm going to talk about RNA. So it's a change of topic compared to the first talk, actually. And basically, what I'll try to tell you about today is what is the meaning of the title here, RNA crosstalk at system level. Basically, every word there has to be explained. But a quick way, yeah, I'm not used to this. So, something that can give you an idea is, I don't know if you've ever heard about this paper, The Strength of Weak Ties. It's a very famous sociological paper by this guy here. This guy. I think it's the most cited paper in sociology overall. It's like 60,000 citations. And basically the idea of this paper is that, well, each of us in our social network, we have some strong ties to those that are really around us, like family and friends, close friends, etc. And as important as these ties are, we depend on the weaker ties that we have with faraway groups in order to gather new information about where we are and how's life going in our community. So that's the strength of weak ties here. Now, of course, biology is full of weak interactions. Like saying that there's lots of weak interactions in biology is a little bit like saying that there's lots of water in beer, right? So it's, but some of them are actually more important than others. And I think I'm going to be talking about one of these examples today. So just to be sure that we're on the same page, I want to speak like a minute about what microRNAs do, what's their job, okay? Now this small piece of RNA here on top is a microRNA, okay? And it's tied to a protein. It's actually just one of the proteins that that it binds to in order to do its job. It's shown here. There's a much bigger complex behind it that is not shown. And this is a long RNA molecule. So this in particular is a messenger RNA. It doesn't have to be a messenger RNA. As long as it's a long enough molecule, it's going to be more or less the same thing. Okay, and the job of microRNAs is the following. They look for binding sites, sequence-specific binding sites on their target, and they bind to them, okay? And once they bind, they basically act as a sort of roadblock for ribosomes. So this purple ball is a ribosome. And if the ribosome stumbles on the microRNA, it can't translate the messenger RNA. Okay? So that's why one speaks about gene silencing. And then, in addition to that, microRNAs can actually trigger the degradation of the messenger RNAs. They don't have to. Sometimes they do, sometimes they don't. I don't think it's entirely clear when they choose to do it and when they don't. But basically that's their job, okay? Silence the expression of genes. Mechanically, literally by blocking the ribosome. On top of this job, so silencing translation, or let me say, along with this job, comes a weird effect that is actually been noticed, I think about 10 years ago. And I'm thinking about the following. So imagine that you have a microRNA there, okay, and its job is silencing genes, so shh. And these are two different messenger RNAs that it can bind. I mean two different messenger RNA species, okay? Now, if for some reason the microRNA, so for some reason I mean physics, okay? If it prefers to bind to this messenger RNA, as opposed to this messenger RNA, every time the cell decides to increase the level of this guy, it will attract messenger RNAs more and leave this guy alone. So an upregulation of this messenger RNA causes an upregulation of this messenger RNA. Okay, there's going to be more functional products of both guys. So there's a positive coupling between these messenger RNAs that is mediated by the microRNA, by the simple fact that they compete to bind the microRNA. Okay? This is what I mean by crosstalk. This is what people mean by crosstalk. This is by no means special to microRNAs or to RNAs. Okay? There's competition for molecules all over the place in biology, but this is, I'm not going to try to, compare this to other cases. There's lots of subtleties. I think one has to be very careful, but so I'm just going to focus on this specific case. So, just to make it a little bit more clear and more quantitative, you can take this this cartoon here and transform it into something a little bit more manageable for people like me, namely boxes and balls and numbers and arrows. Okay, and this is just what each of those arrows in this cartoon represents, a little bit more specifically. So each of these molecules, this is the messenger RNA, this is the microRNA, they can bind to each other, they can unbind with a certain rate. The complex, the microRNA messenger RNA complex can be degraded, and of course each of these molecules can be synthesized and it can be degraded at certain rates. Okay? Once you have this, you can put numbers on these parameters and you can do stochastic simulations. Okay? And what you see is something like this. So imagine these red and blue curves represent the levels of these two species that are competing to bind the same microRNA. Okay? And what I'm doing here is I'm monitoring their level over time. Because this is a stochastic simulation, so imagine Gillespie, there's intrinsic noise affecting both molecules, okay? But where you see the arrow here, I'm doing a perturbation, and the perturbation is just I'm increasing the expression rate, so the synthesis rate of the red molecule. So okay, this is shooting up. And what I see is that the level of the blue molecule also increases. Okay? So this is what I mean by crosstalk. I haven't changed the level of transcription of the blue molecules, only the red, but because they're both regulated by the same microRNA, also the blue one responds when I change the transcription rate of the red guy. Okay? Now this seems easy, but it's actually tricky from a physical viewpoint, and the reason is one of the reasons is shown here. Okay, so this is again the same simulation, but in a setting in which the rate at which the microRNA represses the red molecule is very, very high. So as soon as there is a red molecule, microRNAs just jump all over it. Okay, so you never see a red molecule, basically, a free red molecule, right? And I do the same thing. I still do a small perturbation in the transcription rate of the red molecules. Basically, nothing changes for the red molecule, but I still see the blue molecule that responds. Okay, this is kind of spooky, right? So I don't see any response at all here, but the blue guy shoots up. Okay, this is also crosstalk, but you understand that there's a very, very serious difference between these cases. So to try and make this difference a little bit quantitative, what we tried to do was to describe these responses to perturbation using susceptibilities. Okay? And a susceptibility is just, in this case, tells me what is the change I will see in the level of one messenger RNA if I perturb the transcription rate of another messenger RNA. Okay? So I and J are both messenger RNAs. So I can just calculate it from in these two situations, right? And the difference that you see in the simulations, they translate in very clear difference in how these susceptibilities behave. So in the first case, you can imagine that if I did the same trick on this molecule, so if I perturb these molecules, this would respond in exactly the same way as the blue one responds when I perturb the red molecule. So the two susceptibilities, Chi21 and Chi12, Chi21 meaning I look at the response of messenger RNA 2 when I perturb messenger RNA 1 will be roughly the same. And this is what you see in this plot. So I'm plotting here the susceptibility susceptibilities chi2-1 and chi1-2 as a function of how much microRNA there is in the system, right? But in this case, instead, the two susceptibilities are very, very different. One is very large, so the one where I look at the response of the blue one when I perturb the red one, while the other one is basically non-existent. If I do a perturbation of the blue molecule, the red one will not even notice. Okay. Now this is weird. I mean, this is not totally standard for physics because we're used, especially in statistical physics, we used to think of susceptibilities as symmetric things. Well, in this system, they're strongly not symmetric. We're not. So even though we can look at steady states where we can have some out of equilibrium-like features. Okay,